Carlos, yeah, there you are. So last but definitely not least, the talk we've all been waiting for, Carlos. No pressure. From Champagne Center in Portugal. Carlos is going to be talking about the gourmet fly, dissecting the mechanisms underlying feeding decisions. Please, Carlos, share your screen.
Here we go. Can you see it?
Yes.
Okay, brilliant. The pointer too. Okay. So first of all, thank you very much for your great introduction, Ross. No pressure, and thank you very much, all of you, for taking time to listen to my presentation. So in the time which was given to me, I think I would like to give you a bit of an overview of what are general strategies which we and other people in the field have been using to understand the way the brain interacts with the body to generate behavior. At the beginning, I will give you some kind of vignettes of different approaches which have been taken to understand the interaction between nutrients, physiology, and also the microbiome with behavior. But I'm only going to touch very superficially on the microbiome work because I think Nicole and Adam have done that beautifully, so I don't have to add to that. I'm going to be putting a little bit more emphasis on approaches we use to look at behavior and neuronal circuit, brain activity to look into what's going on in the brain. And I'm also going to try to give you more of an outlook about the future, where MALAB think the field is going, than to kind of dwell too much on the past, which anyway is behind us. So I hope you'll enjoy the ride. Okay, let's see if this is working. Yes, okay. But let me start first by thanking the lab. So this is my fantastic team, actually the ones who do all the work. I'm just their spokesperson. Really a great team of people coming from many different countries and nations. I also would like to thank all our collaborators, which allow us to really do our very interdisciplinary approaches to the question of how nutrition is regulated by the brain. And last but not least, our funders who keep our flies well fed, and obviously also us as researchers. And here I would like to especially thank the Champalimaud Foundation, who's our host, and which is located here. I hope you can see the video. So this is the Champalimaud Center for the Unknown, a relatively recent center on the waterfront of Lisbon, exactly where the ships used to sail out on their discovery trips through the world, and where we have a mix of clinical and basic research on mind, the brain, and also physiology and cancer. And I hope you all come and visit us one day. So when we look at the literature on the brain control of feeding, what most of my colleagues, especially invertebrates, are interested in is how does the brain regulate the motivation of the animal to eat or not to eat. How, especially the hypothalamus, a key region for motivational control in the animal, controls the urge to eat or not to eat. But that's not so much what we would like to understand. So when we look at food, we don't look food as a unitary resource, as something to eat or not to eat, but we look at food in terms of more complex resource, a resource with different nutrients, like carbohydrates, for example, vitamins, fat, proteins, and different types of fats, all types of nutrients which have very different impact on the physiology of the animal. And so what we would like to understand is how the animal regulates its food choices towards different foods with different nutrient contents so that it can optimize its life history traits like aging, something Adam talked about, but also reproduction. And how the animal makes sure that it always has enough nutrients to really be successful, obviously, in evolutionary terms. And we focus mainly on proteins and amino acids. The reason is that while there is quite a lot of understanding nowadays about how the brain measures energy and sugar and carbohydrates, we don't really know how brain in any animal regulates and controls the homeostasis of amino acids and proteins. So we don't know which are the mechanisms by which the brain senses amino acids and how the sensing of amino acids is translated in changes in behavior. What we know is that amino acids are very important, right? They're essential. The same 10 essential amino acids for humans are also essentials for the fly. And this makes it that it's a great system to study because the animal relies exclusively on its drive to eat amino acids to maintain amino acid homeostasis. It can also not properly store amino acids, so we don't have an organ to store amino acids, which means that in extreme cases, what animals start doing is they start digesting their own muscles to regain amino acids, which obviously is not a great strategy. But what we also know is that not only do animals need amino acids, but also that eating too much amino acids is actually bad for animals, from flies to humans, right? So the simple strategy of just over-ingesting amino acids and proteins is also not a good strategy for flies and humans because this will lead to a shortening of lifespan. And this is a fascinating, ethologically relevant position to be in as a neuroscientist if you're interested in decision making. Because the animal has to choose to eat enough amino acids, but not too many amino acids. So it needs to calculate trade-offs. And this is really what you would like to understand in terms of what the brain does in terms of decision making. Okay. So what we would like to really understand is how the brain solves this. How does the brain solve to eat right? What are the computations in the brain which leads the animal to eat right? And how does the brain use information from the body to decide to eat right? So how do you study that in flies? How do you study decision making in flies? How do you now transform this wish to understand nutrition in flies into an assay? Okay. So here, what I'm showing you is a simple behavioral experiment, a choice experiment, like we do in the lab every day with our flies. And here we give the fly two options to choose from. One option, which has a lot of amino acids, which is a simple food spot of yeast. And then we give it another choice, which is a carbohydrate source, which contains mostly sucrose. Okay, so we give these two options to the fly. We have ways to measure how much the fly eats from the two options. I'm going to discuss this later on, but let's just focus here on the results. And what we're looking at here is just how much the fly ate from the protein amino acid rich source, how much the fly ate from the carbohydrate resource, and we have three types of food. Three types of flies. Flies which were kept on a full diet, flies which we have deprived for 3 days of amino acids, and flies which we have deprived of carbohydrates. And what you can immediately see is that the flies deprived of amino acids will selectively increase the intake from the amino acid source, but will not significantly increase the intake from the carbohydrate source. Whereas the flies which lack carbohydrates will not increase the intake from the yeast source, but will increase specifically the intake from the carbohydrate source. So what that means is that the fly is able to sense what the body needs, what it is lacking, and then it's able to transform this need into a specific choice of increasing the intake of the food resource which has that nutrient. And we want to understand how this works. This is not specific for flies, so we know nowadays that all animals, including humans, have protein homeostasis. So if you take humans and you keep them on a low protein diet, you cannot remove completely amino acids from the diet. And then if you give them people who have been kept for 2 weeks on a low protein diet, the choice from different food sources, they will generally over ingest from the protein food. Again, suggesting that they have also protein homeostasis. And we think that this, understanding this phenomenon, is really at the heart of understanding how the brain regulates nutrition, and therefore really also how nutrition is encoded in the brain, in the animal, in the genome, and ultimately what nutrition is and how it is regulated. So as a neuroscientist, however, we think we can also use that to have a profound insight into how circuits and the brain work to make decisions. Okay. So if you look at the history of the recent history of neuroscience, we have learned a lot about how our sensory systems work, about how vision works, how our taste and olfaction works, and how that is transformed then into a motor output. But what we don't really understand so well is how these processes are altered and changed so that computations, the brain makes computations depending on the internal needs of the animal, the goals of the animal to now shape the motor output so that it fits the needs of the animal. And we think that nutrition is a great ethologically relevant and conserved mechanism to start understanding the evolution, but also the functioning of the circuit across time. So the strategy of the lab to tackle this question is to take an integrative, interdisciplinary approach. So we have people in the lab which study the identity and the function of specific neuronal circuits in nutrient decisions. We are interested in understanding what are the molecular mechanisms which allow the brain to sense the availability of nutrients. I'm not going to be talking so much about this today. We obviously care a lot about nutrients and the microbiome, as you heard in the last talks, right? These are key. The microbiome is a key shaper of diet and nutrition. We care a lot about behavior. We care a lot about quantitative behavioral assays. So the key point of the lab, I'm going to be really diving into this today, is to look at behavior in very quantitative, very precise ways. We look at the consequences of the circuit and molecular computations in terms of the output of the animal, in terms of aging and health, reproduction. And also, we look a lot into to which extent the metabolism, the physiology of organs of the periphery talks to the brain to then shape the behavior and the circuit computations of the animal. And we firmly believe that at the intersection of all these different approaches, is where we can gain an insight into the mechanisms which regulate nutrition and therefore also nutritional impacts, the impact of diet on our health. And such an ambitious interdisciplinary mechanistic approach is really something which is almost exclusively possible in invertebrate model systems like the fly. Because here we can do large-scale, non-hypothesis-based approaches, but also we can really manipulate different components like molecules, circuits, neurons, metabolites in very precise ways so that we can really get at the mechanistic implementation of these things and go away from purely correlative approaches as you often have in different systems. Okay. So again, let's think a little bit about internal states and how they might impact brain processing states, right? And here I'm really going to show you some just kind of small vignettes because it's not going to be the main aspect of what I'm going to be talking about today. So I'm just going to show you some things which we have been doing a bit in the lab over the last years. And here really key has been our collaboration with Matt Piper while he was still a postdoc in Linda Partridge's lab and now continuing in his lab now in Monash in Melbourne, Australia, it's been the going away from yeast, which is the main protein source for flies, but which is a very complex nutrient in the system, right? So we cannot really manipulate the content of nutrients of the yeast into a fully chemically defined version of a medium for flies, which we call the olytic medium. So this is a fully chemically defined medium, which has been optimized through painful work by Matt to really be able to replace yeast-based medium in adult flies in a way that the reproductive capacity of adult females, also aging, etc., so all kinds of different physiological readouts are the same in flies kept on this holytic medium and flies kept on yeast. But this now obviously has the advantage that now we can manipulate all the 36 different components in a way that we can really dive deeply into which nutrients give us which effect. This has been key really in allowing us to get insights into how, for example, the microbiome shapes behavior. So as Nicole was saying before, there is a lot of evidence for the fact that the microbiome not only shapes physiology and nutrition, but also behavior. But the way actually that the microbiome does so remains elusive, especially at a mechanistic. And as often in science, we got interested in the impact of the microbiome on behavior through a serendipitous observation of one of my postdocs, Ricardo Leitão Gonçalves. What he had shown is similar to what I've shown you before, is that if you remove a single essential amino acid from the holytic medium, then compared to flies kept on a full diet, the intake of yeast, so the choice of the fly to eat protein, increases dramatically when you remove isoleucine. However, and importantly, this is only observed in flies which do not have a microbiome. If you put back two specific bacteria of the microbiome of flies into the fly, Acetobacter pulmorum, Lactobacillus plantarum, these flies never have a protein appetite, even if they have been deprived of isoleucine for a long time. Importantly, this effect requires the presence of both bacteria, because if you only put Acetobacter or Lactobacillus into the fly, then these animals which only have these single bacteria still show a full and normal protein appetite. And something we wanted to understand is how this works. And for this recently, we have been using metabolomics, especially isotope-resolved metabolomics, which allows us to really observe and follow how metabolites are used by the bacteria to generate new metabolites, which will then change behavior, but also reproduction and other physiological effects. And what we could show, and you can read this in this paper, it's an open access paper, so it's freely available in Nature Communication, is that lactobacillus provide lactate to Acetobacter, which then Acetobacter uses to generate amino acids, which allows now lactobacillus to survive in diets which do not have amino acids, but importantly, also allows Acetobacter to now provide amino acids to the fly so it can keep increased reproductive rate even in foods which do not have amino acids, but also, and we don't understand how this works, generate metabolites which can alter protein appetite. And now we are following up these observations by mapping all the metabolites made in the fly from lactate in Acetobacter and try to identify the metabolites used by Acetobacter to change protein feeding. The other observation which we recently published is that there is a lot of crosstalk among the organs of the fly. So one of the organs, or the organ in the female fly which needs most nutrients, is the germline. Making eggs is nutritionally very expensive. And so something which we found is that the germline not only needs amino acids, but also sugars, especially glucose, to make eggs. And interestingly, the germline is able to tell the brain that the animal should eat a lot of sugars so that it can keep a positive feedback loop, which makes sure that the germline can keep producing eggs. So the germline is able to talk to the brain so that the animal keeps on eating enough sugar so that the germline keeps making enough protein. So there is also crosstalk between organs which allow the brain to know what peripheral but important organs like the germline need in terms of nutrients, so that it keeps providing it to that specific organ. Now, as I said, a key internal state which shapes cravings is reproductive state in females, right? So this is something which most of our female colleagues know. or men know from observing women which are pregnant, is that women which are pregnant have a lot of changes in their feeding behavior and also cravings. And one of the main changes in cravings, for example, in females, is changes and which lead to increases in salt, but also protein preferences. And this is also the case in females. Okay, so if we compare females which are virgin with females which are mated, so mated females here have a filled circle, and virgin females have an empty circle, which have not been protein deprived, which females which have been protein deprived for 3 days, and now we compare the virgin with the mated females, what you can see is while the virgin females also increase protein intake after 3 days of yeast deprivation, it's really mainly the mated female which increase protein cravings, okay? So there's really a strong effect from mating on protein appetite and also salt appetite, something I'm not going to be showing the data for here. So my former graduate student, Samuel Walker, set out to understand how that could be implemented in the brain. And so what has been known is that virgin and mated female flies have very different behaviors, right? Virgin females are receptive, so they are waiting for the male to copulate with them. They retain their eggs, they do not lay their eggs. And something which I was just showing you before and we had shown is that they do not like proteins. But mated females undergo complete change in their behavior when they now become unreceptive, so they're not interested anymore in mating with males. They start laying eggs and they like proteins. And this is mediated, this change in behavior is mediated by a fascinating mechanism which was described by the labs of Marianne Wolfner and Eric Kublé and many, many more. And what's happening is that the male synthesizes a specific peptide called the sex peptide, which is one of many seminal fluid peptides, but this is really the main one important for the changes in behavior, and injects this peptide with a seminal fluid and the sperms into the females. And the sex peptide is sufficient to completely reprogram the behavior of the female. So if you take a virgin female and you inject the sex peptide, the female now, even if still virgin, starts behaving like a mated female. And as a postdoc, I had identified the sex peptide receptor when I was still with Barry Dixon, and we had shown that the sex peptide receptor is in female neurons, senses and detects the presence of this peptide in the uterus, and then sends the information into the brain of the female to change its behavior. And so what people in Barry's lab and also in the lab and you see now the name is failing me, and I'm sure you're going to remember afterwards. It's a great lab in Oxford. Had mapped out is that there are 2 types of neurons which are really important for mediating the changes in behavior. First, there are the sex peptide sensory neurons. So these are neurons which have their dendrites in the uterus and will sense the presence of the sex peptide there. These neurons project to the ventral nerve cord of the female. This is like the dorsal ganglia here for other animals. And there in the ventral nerve cord, you have other neurons, the SAG neurons, which take that information. So they're connected to the sex peptide sensory neurons and sends that information to the brain. Okay, Steve Goodwin, that's the lab. And so what's interesting is that these neurons are always on. So they're always firing, okay? And when they're firing, they make that the female is receptive and does not lay eggs. Now, when you play a trick, because in Drosophila we can play a trick, we can turn off these neurons, or when these neurons get stimulated with a sex peptide upon mating, these neurons are turned off. Okay, so these neurons do not fire anymore, and therefore also the SAG neurons don't fire anymore. And what happens now is that the behavior changes. These females become unreceptive, and they have high egg laying. So they behave as if the female would be mated. So what Samuel did then is he also manipulated these neurons in flies, and for this we can use the GAL4 UAS system. So what we did is we monitored yeast intake of females in which we now turn off these neurons. So what we do is we take a GAL4 line, this is a genetic line which gives us access, genetic access to these sex peptide sensor neurons, and we cross them to a transgene which silences neurons. So this is a potassium inward rectifying channel, which if neurons express it, the neurons go silent, okay? These are the two controls. These flies only have the GAL4 line. These flies only have the silencer. And as you can see in the control flies, you see the virgin mating, nice increase in protein intake in females when they get mated. But now when we turn off the sex peptide sensor neurons and we only turn off these neurons, now the virgins, they behave exactly like the mated females. So we can create a female which thinks that it is actually mated. And we can do the same thing with a different GAL4 line, which now only gives us access to these neurons here. And then when we turn off the activity of these neurons, then these females also think that they're mated. So what we have here is a circuit, a relay of neurons, which brings in the mating information from the female uterus to the brain and completely changes the behavior of the animal. Because now these females will start increasing their intake of yeast and sodium, and that's really important to boost the egg production, which is happening in these animals. So what I've shown you now in these last 15 minutes is different examples of internal states, internal nutritional states, metabolic states, but also mating states, reproductive states, but also microbial states, which all seem to change the way the brain works and the brain mediates changes in appetite. Okay? So now we would like to understand how these mechanisms of nutrient sensing and of interaction of different organs act on the brain to change behavior. So how are we going to do that, right? I mean, it's not like we can ask the fly, hey, what do you like to eat? Hey, fly, how do you feel like, right? So we need to rely on behavior, right? And so here, we're going to be relying on very precise and quantitative dissections of behaviors. I'm going to show you why that really matters to understand how specific neurons work. And I'm going to start by looking at the actual eating behavior of the animal, the so-called feeding microstructure. And I'm not going to go into the details, but we have shown that the way the feeding motor program works in flies is very similar to how it works in rodents and even in humans in terms of chewing, but flies obviously don't chew. But what flies do is they use their proboscis to eat, okay? So this is a fly here eating on a yeast spot. And what you see here on the head is its proboscis. And you see that it's doing this very, very, very fast kind of eating behavior. And it's also exploring the yeast spot to try to find the place where there is the best type of yeast, and obviously also the best type of microbes for their microbiome. But let's look a bit more precisely, and I really hope that you can get to see these videos in high quality. Let's have a look at the high resolution, slowed down analysis of their feeding motor program. And what you're seeing is that the fly has a very, very stereotyped feeding behavior where it extends and retracts the proboscis and also the labellum opening in a very, very stereotyped way. And so we set out to measure that in an automated way. And this was a collaboration between my former postdoc, Pavel Itzkov, with Michael Dickinson at that time at the University of Washington and now at Caltech. And the idea we had was to use touchscreen technology. So some of you might have electronic devices like phones or iPads or screens, which work by measuring the touch of your finger on the device. And so our idea was not to measure the touch of the fly with a device, with a screen, but the touch of the fly with a food spot. And the beauty of this is that this type of touchscreen technology works with sensors which measure changes in capacitance, okay? And what we're going to be using is exactly the same type of sensors which you find in different types of mobile phones, and because they're mass produced, one of these sensors is very cheap. It costs around €2, but it is also very sensitive because they have been really perfectioned to the use in our pockets. And so the idea is that we can maybe measure the touch of the fly proboscis with the food, and that might give us access to the feeding motor program of the fly. So this system, we call it the fly path, and it consists of two food ports or channels. In one, we can put, for example, yeast or protein-rich food or amino acid-rich food. And in the other, we can put, for example, carbohydrate-rich food like sucrose or apple jelly or whatever type of food you would like to choose. You would like to have the fly choose from, sorry. Then you put one fly here, you close it, and then when the fly eats from one of these channels, it's going to be measured by this sensor here, which does the capacitance to digital converter. This is connected via USB then to the computers, and we can have hundreds of these in the lab to choose, to measure now the choice of the fly and the feeding behavior of the fly from two different options. So this is how it looks. So here you have a fly eating from a fly pad food port. Here you see it standing on one of the sensors of one of the electrodes. The other electrode is in the food. And here you see now the signal in the capacitance signal of the fly. And what you see is that we have this very, very stereotyped pattern which corresponds exactly to the rhythm of the fly when it's eating. So we can write now a program to just extract now every single one of these bumps, and these bumps we're going to call them a sip. And we then showed that one of these sips, which you can imagine like a bite, right, when you take some food, corresponds pretty exactly to a 1 nanoliter of food intake in the fly. And so by counting the numbers of these bumps, of these sips, we can extrapolate how much the fly eats over a certain time automatically. But not only that, and now it's where it gets really interesting for us as neuroscientists, we can look at how the behavior is organized. Sorry, let me take a sip of water. So if you think about behavior, especially in freely behaving animals, it might look very, very puzzling how you're going to make sense out of that, of animals walking around and making different choices and trying to understand how this is controlled by the brain. So what we need to do is we need to reduce this complexity into specific variables which allow us to explain the behavior as completely as possible, and then look into how these variables are controlled by the brain. And in terms of motor output and motor pattern, actually it turns out that most of our motor behavior is very stereotyped, right? Like when you walk, like it's not like you walk, you think, you walk, you think, you take a step, you think, you start walking, and then you take multiple steps. And so motor outputs are organized in bursts normally, okay? And so normally these two variables which regulate how far you're gonna walk, or in this case, how much you're gonna eat, which is how long the bursts of eating are in the case of flies. In your case, how long the burst is of the food you're gonna be taking into your mouth. And then the second variable is how long the interval between these bursts are gonna be. In human cases, how long it's gonna take until you start taking again food. And so it turns actually out that when you starve an animal, what's happening is that the bursts start becoming longer and longer, which means that the flies will be eating more and more, but also that the interval between the bursts start becoming shorter and shorter and shorter. So now the question of how animals increase their intake when they are starved or deprived is turned around into us asking two specific questions. How does the brain regulate the length of a feeding burst? And how does the brain regulate the length or the shortening of an interval of this burst? Okay. And so the way this works and what are the neurons which are regulating this was again work of my former student Samuel Walker and two postdocs, Katrin Steck and Pavel Itzko. And what they found is that this is controlled mainly by the taste system of the fly. Okay. So the tip of the proboscis is called the labellum. Okay, you can think of it as the tongue of the fly. And this has different taste neurons on there, like for us, right? And then these neurons in insects come in two ways. They come as sensilla, so these neurons are inside of hairs which are on the tongue of the fly. But there's also a special type of neurons which are called taste pegs, and these are inside of the labellum of the fly. And so we got really interested in these taste pegs, which you see labeled here with a specific, again, genetic line. So this is a genetic line which allows us to label these neurons which are inside of the labellum, so of the tongue of the fly, and which are taste neurons, right? And which people had not really associated with any type of feeding behaviors. But what Katrin and Samuel and Pasha thought is that these could be really important for mediating yeast feeding. And the way we're going to actually analyze what they do is using optogenetics. So optogenetics is another revolution in neuroscience lately, right? And so these are channels, genetically encoded channels, which will make a neuron fire or which will make a neuron become silent when light is turned on. So you can express these channels, these optogenetic channels, so-called opsins, on neurons using specific alpha lines. And now when you turn on the light, either that neuron is going to fire or that neuron is going to turn off. And now what we're going to do is we're going to use a trick, and we're going to now turn the light only off after the fly has started to eat. Okay? And so the theory was that these neurons are specifically required to regulate the length of the feeding burst. And indeed, if you look at the length of a feeding burst in animals which have not been, someone is painting here on my presentation. If you look at the length of a feeding burst in animals which are not getting light and are expressing now an inhibitory opsin in these taste neurons, you see that they are around 20 sips in one burst. But when we turn on the light after the fly has had 3 sips, now the fly stops eating. And now you have shortened the length of these bursts, which means that these taste peg neurons are specifically required to actually control the length of the feeding burst, okay? So which means that taste is not only important for the fly to perceive what it's eating, to know what it's eating, but that taste neurons are also important for regulating motor control, motor output of the animal, in this case specifically feeding. Okay? So now let's start to look into the brain. Okay? So now a question is, do these neurons actually detect protein-rich food? Do these neurons really sense yeast? Okay? So the question is then, how can we watch the brain working during decisions? And so now we're going to start to be looking, into how the brain works. Okay? So how could we do that? So here a very important discovery was the use of a molecule called GCAMP. So GCAMP is a modification of GFP, where GFP was changed by adding calmodulin calcium binding domain. Normally, GCAMP does not fluoresce when it's excited, but when a neuron fires, then calcium binds to GCAMP, and now this molecule becomes bright. Which means that GCAMP allows us to watch neurons working, because if neurons are spiking, are becoming active, then when we express the sensor in a specific neuron, then this neuron should turn green. Okay? So what do we need for that? We need a special microscope, and we need a very good postdoc who knows how to open the cuticle over the brain of the fly so that the fly is still alive. Okay, so the fly is going to be alive. The fly is going to be placed under the objective of this microscope, and now we're going to be expressing GCAMP in special neurons in the brain of the fly. And we're going to be stimulating the labellum, the taste organs of this fly, which is still alive, right? So as much as we can say happily alive, under this microscope, and we're going to look at how these neurons work, okay? This is how such a microscope actually looks like. Okay, so this is the objective of our 2-photon microscope. Here, we're coming with the food. Here, you see the fly. Here's a actually zoom in here. Okay, so here's the fly on the objective, and here we're zooming in even more. So here is the fly alive under the microscope with an opened cuticle, so we can look into the brain. And here we're going to be coming and putting the taste on the fly tongue. And this is going to be done by observing it with a camera, so we're really sure that we are coming there with the food. Okay. And so now let's look at the brain where it's working. Okay. So now we're going to be looking at the part of the Drosophila brain where these yeast, these protein taste neurons are coming in. And we're going to be making a movie and looking at the fluorescence inside of these projections in the brain of the taste neurons when the fly eats yeast. So now we put yeast on there, and what you see is, bam, these neurons start firing, and when we remove it, it goes down again. So let's repeat this. And you can see their neurons firing, okay? And so this gives us now access to the activity of these neurons in a living brain. And we can look at how the brain reacts to foods, right? So these neurons turn actually out to be really important for detecting yeast, something I just showed you, but also to control the initiation and the sustaining of yeast intake. And also they're especially important for yeast sensing because they don't really react to sucrose or to water. Okay. So with this, we can start understanding how now this sensory information is turned into specific motor output in terms of the feeding mechanisms. And now in my last 5 minutes, I really want to show you a little bit of the future of where we're going now with this, right? Because we would like to understand what changes in the brain when nutrient appetites change. And so this is work now from a current postdoc in the lab, Daniel Münch, who is our imaging expert. And again, we're going to be looking into how the brain works when the fly is eating. And we're going to be looking specifically in this part here of the Drosophila brain. So in gray here, for the ones who have never seen it, this is a 3D rendering of a Drosophila brain. And here in the ventral region of the brain is the region where the taste information comes in, the gustatory input, and also where the feeding motor output is. So we think that this zone is enough to understand also which happens in the brain. But now we're not going to be looking at activity of one neuron at a time using specific GAL4s, but now we're going to do something which is really kind of state which we have been developing, which is to now image all neurons in the brain. Okay, so we're going to be expressing GCAM, this calcium sensor, which allows us to see the brain active in all the neurons in the brain. And we're going to do that in flies which are starved, which are protein deprived, which are mated, which are virgins, to see how now the activity of the brain changes when the fly tastes food in different internal states. Okay. Again, this is how the prep looks like. And this is now really the activity of the whole brain in 3D, because we have a special microscope which is very, very fast. So we're going to be looking at the activity of the whole ventral part of the brain when the fly eats yeast. Okay. So what you see here, when the stimulus comes on, that means is when the fly is eating yeast, we see now that there are special parts of the brain, neurons, firing now, and yet that you see in terms of these yellow spots. These are again the taste neurons, the ones you already know. You're all now imaging experts, right? And now when we go more dorsal, we start seeing new neurons, which we do not really understand and which are now firing when the animal is eating yeast. And so now this gives us access to understand how the whole brain processes information. And we can also compare the brains of animals which are fully fed with animals which are protein deprived. And as you can see, there are drastic differences between the brains of animals which are fully fed with animals which lack proteins in terms of the brain activity when when the fly is eating. And now we're using different mathematical ways to understand how the activity in all these different regions and all these different neurons are changed when the animal is starved or not starved. And so what we can see is that indeed the metabolic state of the animal, so when the animal is lacking specific amino acids, then actually there are brain regions like in the projection of the taste bud gustatory neurons, which when the fly is deprived, now becomes more active. And you don't have that when the fly eats sucrose or when the fly drinks water. So indeed, what the internal nutrient needs of the animal seems to be doing is it seems to be enhancing the perception, the chemosensory taste of food when the animal is lacking that nutrient. And we can now map the different neurons and the different parts of the brain where that is happening, and we can map that then to specific changes in activity. And again, this is specific for protein feeding because we don't see that for when the fly is, for example, eating sucrose, okay? And a big question which we're trying to address now is how does the mating state affect this gustatory processing? But this is really going on state. The experiments are being done now, I hope. And I hope next time we talk, I can tell you how mating changes information processing in the brain. Okay. So this allows us now to really couple internal state changes to changes in behavior, in brain processing and in behavior. And for the sake of time now, I'm going to skip how we're looking at foraging and go really to the end of my talk. Let me now switch here. Okay. So what I've been telling you today is that nutrients change processes in the brain within the animal physiology, which allows the brain to know what is the internal state of the animal, changes the way the brain processes sensory information and generates motor output to shape different aspects of feeding behavior, the feeding exploitatory aspect itself, but also more complex foraging, food search type of behaviors, which I didn't have time to discuss with you today. And this therefore gives us access to understanding how nutrients affect behavior, but also more generally, how decision-making is encoded in the brain. And this has many, many different important implications also when we think about health and well-being of humans, because many, many diseases are actually using female insects as vectors for being transmitted. And most of these diseases are transmitted by female insects, which are looking for proteins and to make sure that they can sustain egg production and therefore reproduction. And this is all mediated by changes here in this tiny brain, which is driving these females to sting humans and therefore transmit viruses and bacteria, which make them people sick. And also many agricultural pests rely on changes in feeding behavior in insects. And so in understanding insect feeding behavior is paramount to the well-being of humans. But, and here is really I would like to end with this, I think as scientists, we shouldn't forget that also understanding these processes give us access to beauty in nature, okay? So this is this picture here is an amazing phenomenon. These are male butterflies which will go and drink from the tears of turtles. This is called lacryphagy. And the reason these male butterflies are drinking from the tears of these turtles is because tears have a lot of salt. And so these males are taking the salt from these tears and then giving it to the females when they mate with them as a nuptial gift to increase the reproductive output of the females. And it's really this phenomenon, this nutritional behavioral phenomenon, which gives rise to this beautiful picture, which we can see in nature. And I think reminds us that not everything is about using the knowledge we gain, but also about understanding the beauty around us. This is the team which does all the work with us. And we are always looking for talented scientists to join us. This is the view from our fly facility and the fly station. And I also invite you to follow our brain-body interaction virtual seminars, which we host every Monday, where we invite a speaker to discuss the interactions between physiology, gut, microbiome, other physiological organ systems with the brain, and try to think about how we can understand behavior in a more big picture way. Thank you very much.
Carlos, that was beautiful. I love the way you ended that as well. Very nice. And I suspect you've got some student and postdoc candidates on the chat, so you may okay, so let's ask some questions, and then before we wrap up, and there's quite a few, so I'm again going to have to pare them down. Miriam Adeniki Saladin.
Ross, give us the view of the participants, please. We are still with the slideshow. Yeah, thank you.
I'm just
That's from Carlos. Yeah, you have to. So actually, I don't know where Miriam is. You're welcome to ask the question. Otherwise, I will. So Miriam, oh, hang on, I may have to do a little muting here. We're good?
Please go ahead and ask the question on my behalf.
Okay. Although my voice will not be as cool as that for sure. Miriam wants to know, there's a few comments about the fly pad setup, how great that is. It's pretty amazing. She describes it as a different level of awesomeness. I don't know, I think it's right in that level of awesomeness.
Thank you very much.
Can it be adapted for neurobehavioral studies? And is it, and I guess this is going to go out to the community, is this available in, for example, Nigeria?
So yeah, so we have made all the software, but also the hardware designs publicly available. So it's all open source. If you go to our webpage, so we're just revamping our webpage of the lab, but you should be, you should be having access to all the designs on our GitHub repository of the lab. Also for the Octopad and also for all our other things. So we try to do as much as possible open access, so you can build them yourself wherever you are. And I mean, it's not super complex, or you need to know how to do some soldering. And if you're at that level, you should be able to build it up.
Okay, cool. And okay, people, there are way too many questions here. You're gonna have to go into Slack for these. So let me ask a few.
Okay.
I'm also the muter-in-chief here. More questions. So Lola has several questions, which I'll sort of sprinkle them in. How long does the fly keep the ability to know what it needs?
Okay, so that's a great question. So in terms of the mating state, we understand that relatively well, because but that's really from work done, especially by Marion Wolfner, on how the sex peptide works. So the sex peptide is bound to the sperm and is then injected into the female, which means more or less that as long as the female has sperm, it's gonna have a change in the post-mating response, and therefore in the kind of preferences. As soon as the sperm disappears, there's no sex peptide there anymore, and the circuit becomes active again, and then the the post-mating response goes back to the virgin state. In terms of the nutritional state, the most parsimonious explanation is that the animal is measuring amino acid content in the hemolymph, and then is able to kind of really predict and approximate the nutritional needs of the animal. And we have cloned a gene which we call beef eater, which we found in a in a forward genetic screen, which if the fly is mutant in this gene, it will have a constitutively high protein appetite. Interestingly, it's fully conserved gene. It's only expressed in neurons, and we think it's part of kind of a conserved nutrient sensing mechanism used by the brain to regulate protein appetite. Therefore, also the name beef eater.
Nice. Priscilla Akayo wants to know if the activity of the sex peptide wears off. For example, if you just mate the female just once, how long does it take for this preference to last? How long does it last?
And this is correlated with the availability of sperm. And when the sperm disappears, sex peptide disappears from the system, and then the female goes back into a virgin state.
Pretty much immediately.
Well, it takes some days, right? Because females keep sperms from some days. So it doesn't go away immediately, actually.
Sure, sure. Tulup Agambadidi, again, I apologize for my pronunciations. I want to know, how do we identify a yeast that's gone bad?
Sorry, I
How do we identify a yeast that's gone bad?
So flies are very good actually at detecting spoiled food, and the way it actually does that is using olfaction. So there is a series of very nice papers showing that contaminated yeast, so when the food has bad bacteria, for example, growing there is an odor called jasmine being produced then, and that's then sensed by specific smell neurons on the antenna of the fly, and that is then highly aversive for the fly, so the fly does not really eat that.
Got it. Mariam Saladin wants to know if you've tried microelectrodes to study the brain activity during the fly decision instead of the more complex cyclic AMP.
Yeah, I mean, some people can do electrophysiology in living, behaving flies. That's, however, much more complex for the brain neurons than it is to do calcium imaging. Let's say there are different challenges, right? Because you have to find the neuron you're interested in. So we have colleagues here at the Institute, especially in the lab of Eugenia Chape, which are amazingly good at that. But we stick with the imaging because also it gives us a more global picture of what's going on. But indeed, now that we are following up on activity of specific brain neurons involved in some specific aspects of decision, we would like to go and really do patch clamping. But that's really very challenging technically to do.
Got it. You know, there are a bunch more questions, so I'm gonna pull the plug here, and please check out the chat, which is quite a few. And I know, Sophia, you've been putting some of them on Slack, much appreciated. So we're gonna call this a day. We've got 2 more days and different topics. So there's gonna be a pretty broad sweep. So I hope, like me, I can remember how to do this. A little bit of applause for our speakers. I think would be appreciated for them taking their time and sharing their science. It's much obliged to everybody. And thank you everybody who has participated in, especially those who've had questions. Those questions have been awesome. I will see everybody tomorrow.